So this is lab number 10, part A, DNA isolation. And this particular experiment will require some items from your lab kit, so I'll show this to you in a moment. But what we're going to be doing is we're going to be isolating DNA from unprocessed wheat germ. So we're going to talk about how we're going to do that, and you do want to understand the importance of each step and why we do each step, what the reason is. So first off, remember that DNA is stored within the nucleus of the cell, right, when we're talking about eukaryotic cells. So it's stored within the nucleus. So we have to actually get inside the cell and inside the nucleus to be able to extract and get at that DNA. So there are going to be processes that we're going to do that will allow for that to happen. When you look at the bottom of your screen, right here, you see that there are 3 main steps involved in isolating DNA. So the first step is to lyse the cells to release the DNA. So the term lyse means to split. So we have to split that cell open so that we can get at the DNA which is stored inside. The next step is to separate the DNA from the protein surrounding it and any other cellular debris. And then this third step is to isolate concentrated DNA. Because if you were just to take DNA from a single cell, you wouldn't be able to see it. But if we concentrate a lot of DNA from a lot of different cells, a bunch of cells rather, then you can see it. Will clump up and it'll be visible because you pulled it from a large source. So that's what we're going to do. So we're going to talk about those steps. I'm going to go ahead and scroll on your document for you here. So you're going to actually make what's called an extraction buffer. And the extraction buffer is used to break open or lyse the cell, remove proteins, carbohydrates, and other cell parts. We're also going to dissolve the DNA into solution. So the buffer that you're going to make will consist of a liquid detergent, any kind of, just dishwashing soap is what we're talking about. So any kind of dishwashing soap would be great. You're going to also include salt, and you're going to have water. And that's going to make your extraction buffer. So what the detergent does, how you use detergent, dishwashing soap when you clean dishes so that it gets rid of the fat or any of the proteins and carbohydrates that you have on your plate? It's the same kind of thing. You're going to use that soap to disrupt the cell membrane and to isolate the proteins and carbohydrates so that we can remove that material. First off, we can get into the cell, and then we can remove that material from the DNA. So again, the detergent's going to dissolve the plasma membrane of the cell. It's going to bind to proteins and carbohydrates so that they will precipitate out into the buffer and we get them away from the DNA itself. Okay, so let me scroll a bit more. So here we are now. What we're also going to do is we are going to heat this mixture up, and what the heat will do is it will denature any enzymes that might potentially break the DNA down. So it's going to denature DNase enzymes that would interact with the DNA and break it down. So we want to heat it up to do that. And then we're going to isolate it using very cold ethanol. So I'll talk about that step when we get to it, but you do want to freeze the ethanol for a while, and it's rubbing alcohol is what it is. But it does need to be really cold in order for this to work. So when I have had students that have sent pictures of the extraction and they said, well, it didn't really work, I kind of come back to that step. That really needs to be cold. And you do need to watch the temperature of your heat that you apply as well. So the temperature involved in this experiment is actually pretty critical. If you want to get good results, you do need to pay attention to that. So DNA, the deal is that DNA is not soluble in that alcohol layer. It's soluble in water, but not in the alcohol layer. So it will pull up into that alcohol layer and form a blob, because it's kind of trying to pull in on itself to stay away from the alcohol, and it'll form a big globby ball that you'll see on the top, and that will be your DNA. Okay, so here are the materials that you're going to need. You're going to need your thermometer, because like I said, the temperature issue, let me see, can you see it? There we go. I'm in the bottom right corner there. Your thermometer, because the temperature part of this experiment is pretty critical. You also need your wood applicator stick, and the wood applicator stick is attached to the Kit 7. So, ooh, there we go. Right there. The Kit 7 bag. On the back, you have a little wooden splint taped to the back. Okay, and then in the bag, that is the wheat germ. Okay, so that's the pre-measured raw wheat germ in the bottom of that bag. You will need 2 conical tubes. You want the 15 mil that you're used to using. You've used it before. That's your 15 mil conical tube. But then you also need a 50 mil. And the 50 mil conical tube is this guy right here. Okay. All right. You will also need a pipette. And the pipette is this guy. So this is your pipette that you're going to use. Okay, now you're going to provide a measuring spoon, a timer, ice, or access to a freezer. You're going to have tap water heated between 50 and 60 degrees Celsius. You need table salt, you need liquid detergent, and isopropyl alcohol, 91% isopropyl alcohol. So what that is rubbing alcohol, that you can get at Walmart, you can get in pharmacy, supermarket, wherever. 70% will work, but 91% works best. So if you can get the 91%, that would be better. Okay, so looking at the procedure, you're going to pour that pre-measured wheat germ into your 50 mil, the large one, your 50 mil conical tube. You're going to pour 14 mils of that rubbing alcohol into your smaller tube, into your 15 mil conical tube right here. And you're going to place it in your freezer, or if you don't have a freezer, you're going to just plunge it into some ice. Okay, then you're going to heat approximately ½ a cup of tap water to between 50 and 60 degrees Celsius. You can do that on the stovetop or using a microwave. Be sure to use your thermometer to measure that temperature because you do want it to be between 50 and 60 degrees Celsius. Okay, you're then going to add that heated water to the wheat germ in your big tube, right, this guy. You're going to add that water to it to bring the total volume to 20 mils. Add a pinch of table salt. Stir for 3 minutes using that wood applicator stick. Then add 1/4 of a teaspoon of your liquid detergent and mix gently every minute for 5 minutes. So you do want to stir it, gently and slowly. Try not to create foam or damage the DNA by mixing it too hard. Okay, your extraction buffer is now complete. Let me go ahead and pull this up. You are then going to place the 50 mil conical tube containing that wheat germ and extraction buffer that you just created with the detergent and the salt. You're going to place it in the freezer or put it on ice for 5 minutes. So do set your timer. At that point, then you're going to remove both the 50 mil that has all your stuff in it, right? And you're going to remove that 15 mil conical tube that has your isopropyl alcohol in it. You're going to remove that from both of them from the freezer or the ice, wherever you had it. Okay, then it says to tilt that 50 mil conical tube containing the wheat germ mixture and slowly pour the cold alcohol down the side of the tube. So you're decanting, if you will. So you're going to be tilting and slowly adding that ice cold ethanol so it layers. You want to layer on the top of your solution. And you have a picture there, figure 3, right here, that shows how that alcohol should layer on the top. Okay, but you do have to pour it in slowly so you don't mix the 2 layers together. All right, so then I'm here at number 11. DNA first precipitates at the water alcohol interface. So what that means is where the alcohol should be layered on the top, and then you'll have the extraction buffer with the DNA layered below that. So right where the alcohol meets the extraction buffer, that's called the interface. That DNA will start to precipitate at that interface up into that alcohol layer. So what you'll see when you watch your tube is you should see bubbles start to form. Bubbles will begin to form at the interface, and then you'll see slowly this globby, mucousy looking stuff pulling up into that ethanol layer. That's how it should work. So let's see, it says wait 15 minutes. right, to get all the DNA accumulated, and then gently pull up that mucus-like material, which is your DNA, that's the DNA from the wheat germ, with your wood applicator stick. So you take that wood applicator stick and you kind of swirl it. So here's my stick. So you're going to be kind of doing this. You're going to be kind of swirling it, you know, like cotton candy, like you're trying to get that DNA to grip onto the end. And then you should see a big mucousy glob at the end of your stick. Okay. So you will then take a picture of your tube. It says, okay, well, take a picture. Okay. And answer your lab questions. It says, if you wish, you can store the DNA by removing the DNA from that conical tube to the 15 ml and then add enough isopropyl alcohol to cover. So if you want to do that, you can. Be sure and include a picture of your results, please, so I can see, you know, kind of how it went. From there, let me scroll. You're going to go to Part B, which is looking at electrophoresis. So you do want to do some reading in your textbook before you get to this. I'm also going to post another little explanation for you on electrophoresis. I'm not going to include it in this because I'd like to draw it out and include it as a separate video so you understand the process behind DNA gel electrophoresis. So I'll go ahead and just say a couple more things and end this for you, because I do want to go ahead and again make another video for you over electrophoresis. But for the procedure that you'll do for part 2 is you click on that link that you see right here, right? You're going to go to that link, perform the virtual lab, and learn about electrophoresis. When you're finished, answer the questions about it. It says, FYI, you will be asked to submit the sizes of your bands. So you're going to want to record that or write that information down as well. Okay. So this is a great lab. If you have children, get them involved in the actual DNA isolation because they love it. It's really a fun thing to do, and it works out really well most of the time. It's kind of foolproof. All right, well, have fun. Look for the other video that I will also post over electrophoresis.